Okay, very good morning to one and all. Very warm welcome to all of you. I am Dr. Ramanlal Narayan Kachwe, Professor, Department of Pharmaceutical Chemistry, Sir Dr. M.S. Gosavi, College of Pharmaceutical Education, Research, and Nasik. As you already know, we have organized one day, 1 week international e-FDP and e-STP on empowering pharmacy education, navigating regulations, global trends, and advancing outcome based education and research. For this, we have a wonderful personality, Dr. Sougata Ghosh, Associate Professor, Faculty of Science, Kasetsart University, Thailand. I take this opportunity to welcome today's guest, Dr. Ghosh, an expert in nanopharmaceutical and integrated and interdisciplinary green approach for this online EFDP. It's my great privilege to introduce to our second day first session speaker of the 1 week international EFDP and ESTP, Dr. Ghosh sir. Sir has huge research experience in that. Dr. Sougata Ghosh is currently working as a visiting professor at Department of Physics, Faculty of Science, Kasetsart University, Bangkok, Thailand. Sir is associate professor in the Department of Microbiology, School of Science, RK University, Rajkot, Gujarat. He is active researcher in nanomedicine who is obtained his bachelor degree, masters and PhD in microbiology from Savitribai Phule Pune University, India. He was a postdoctoral fellow at Northern University, USA. He has served as a resource person and invited speaker in 160 international and national conference, symposia, workshop in various countries. As like that, most notable is ICMR 2023 at Cebu Normal University, Philippines, JICEST 2023 at University of Jambi, Indonesia, Mexico, Harvard University, etc. Sir has associated editor of Frontiers in Chemistry, currently current microbiology, Springer, and member of editorial board, reviewer team of 101 international journals like Nanoscale, Journal of Phytochemistry and Phytobiology, uh, Journal of Diabetes Research, International Journals of Advance in Engineering Research and others. He was a principal investigator of Department of Biotechnology, Government of India, funded Folds project and had conducted numerous workshops in various colleges and universities throughout India. He has received several prestigious awards like Junior Research Fellow in 2011, Senior Research Fellow 2012, DST Overseas Visiting Fellowship 2019 in Nano Science and Technology, Reviving University Program Fellowships 2021-22, Bharat Gaurav Puraskar 2024. Sir has filed and patent 271 publication, 8 patents and 271 publications that include the 10 books and 168 book chapters, 93 international research and review articles. He cited, he got the 5,550 citation. H index is a 40 and I-10 index is a 87. He listed among the world top 2% scientists for 2024, 2023, 2022, released by the Stanford University, USA, and elsewhere. He has published highly cited international journals like International Journal of Nanomedicine, Journal of Nanobiotechnology, Journal of Nanomaterial, Journal of Nanoscience and Nanotechnology. We have such eminent, renowned and dynamic personality with us. Now I request Dr. Ghosh sir, please guide the delegates on the topic of nanopharmaceutical and integrated and interdisciplinary green approach. I am sure we will be enriched by this session. Once again, I welcome all of you for this session. Thank you, sir.
Thank you so much, sir, for the kind introduction. So I will share my screen now. Okay. So hope it is visible, full screen. Yes.
Yes, yes.
Okay. So first of all, I thank the organizers of this wonderful international e-FDP and e-STP. So today's topic, as you can see, it is nanopharmaceuticals, an integrated and interdisciplinary green approach. What we are doing over here is to develop new nanomedicine. And in order to develop this new nanomedicine, we can integrate all possible disciplines of science. So it is no more science in isolation. Now we are trying to move towards many branches of science working together to get the nanomedicine. So as you can see, the history of nanotechnology. So why we are talking about nanotechnology? Why nanomedicine? Why nanopharmaceuticals? It was Sir Richard Feynman who told in like in 1959 that there is plenty of room at the bottom. So that means when we actually go down, when we actually slowly cut down the particles into smaller and smaller and smaller form or dimensions, then we get completely different properties. The physicochemical properties changes, the optoelectronic property changes. So that's why if we try to make material very small, then whatever the bulk material is showing, that property is completely different in nanoform. So he got the Nobel Prize in Physics in 1965. So this was a groundbreaking research. Now the question is, what are nano? What are the nanoparticles? What are the nanodimensions? The particles which are ranging from 1 to 100 nanometer, they are called nanoparticles. How we can synthesize the nanoparticles? So here you can see in the left hand side, you have very bigger bulk particle. Now you break it down into smaller, and then you further break it down into more smaller particles. So when you make these very small particles, which are in 1 to 100 nanometer, then we get this nanoparticle. So from bigger to smaller, this process is called top-down approach. This is one approach of synthesis of nanoparticles. There is another approach of synthesis for the synthesis of nanoparticles. That is, initially you form very small particles, which is called nuclei particles. And then they grow. There is particle growth. And then you get these greener particles. And then these particles become larger, further growth in the particle dimension, and you get the particle which are 1 to 100 nanometer. So this from very small to nano dimension is called bottom up approach. So these are the two approaches. One is top down approach and another is bottom up approach. Now the question is, do we get these nanoparticles only in the lab or we can get in the nature also? Now it is very important to note that even in the nature, we have such nanoparticles. So over here, if you are looking at this butterfly, this is called Bistonina biston. It is a male butterfly. The scientific name is Bistonina biston. Now, if you are looking at this butterfly wings, you can see there are four different colors. There is something brown in the over here, which is made up of again light brown and dark brown. So this brown color part, it is called androconial organ of the butterfly. So it helps in attracting the female butterflies. It releases pheromones and it helps in attracting the female butterfly. This brown color androconial organ is made up of light brown scent pad and dark brown scent patch. So these are two parts. Here you can see the black part and over here you can see the blue part. Now we might think that these wings have these colors, brown, black, blue, but actually when you see the microstructure of the wings, you will see there are different patterns over here. There are scales. These scales are forming certain patterns. The scales with the circular hole in between, and they have the fibrous structures. They form this dark brown color. The scales over here, which are having fibrous structure like this, they form the light brown color. When the pattern is like this, they will form black color. When the pattern is like this, they will form the blue color. That means these structures are acting like nanoprisms. When the light is passing through these nanoprisms, then there is diffraction. And because of this dispersion of light, you can see different colors. But actually, there is no color in the wings of the butterfly. So this process is called structural colors, where the change in the dimension of the scales give you different types of colors. These are called structural colors. Now, what are the use of these nanoparticles? What are the applications of these nanoparticles? These nanoparticles you can use in electronics, semiconductors, catalysts, biosensors, solar cells, and different others, even medicines. And very recently, they are used largely in nanomedicines and drug delivery. So, what we are doing in our lab, we are synthesizing different types of nanomedicines. These are made from different types of nanoparticles. And these nanoparticles are not synthesized chemically or physically. These nanoparticles are exclusively synthesized biologically. There is biogenic nanoparticles, and these are green synthesis approach. So what we are doing over here, you can see this structure, very small spherical nanoparticles, triangular nanoparticles, hexagons, these are gold nanoparticles. These circular or spherical nanoparticles and hexagonal nanoparticles, these are silver nanoparticles. These are core shell bimetallic nanoparticles, gold and silver together, bimetallic nanoparticles. These are copper nanoparticles. These are magnetic nanoparticles. These, you can see iron oxide is in the center. These are gold nanobeads. These are liposomal nanoparticles, palladium nanoparticles, and surface defect rich zinc oxide nanoparticles. So we are synthesizing different types. I will be discussing only a few of them today. So these are the nanoparticles, and our research on nanomedicine, they are funded by CSIR, DBT, Foldscope Instruments, Stanford University, US and Thailand. Now, what is nanopharmaceuticals? Nanopharmaceutical means when you are cutting down the size of the medical products, the pharmaceutical products, into nano dimensions. They are very small. So these are called nanopharmaceuticals. Now, this is very interesting plant. It is called Dioscorea bulbifera. I stayed in Maharashtra for 15 years. So that's why I did my research on different endemic plants of Maharashtra. And then I shifted to Gujarat. I did some research on the different plants of Gujarat and then US. And now we are doing on endemic plants using endemic plants of Thailand. So different parts of the world, we are taking different plants and then trying to explore their nanobiotechnological potential. So this plant is widely available, widely found in Maharashtra. This is called Varahi Kand. And these plants we found in Jawahar region of Nasik. So once I was traveling to Jawahar region of Nasik and I found this plant which is called Dioscorea bulbifera, Varahi Kand. So you can see one Dioscorea bulbifera tuber, only a single plant can have so many different types of therapeutic potential importance. It can be used as antibacterial, antifungal, anti-tumor, any kind of disease you take the name of, and you will see those can be cured by Dioscorea bulbifera. So Dioscorea bulbifera is such an important medicinal plant. Now, first thing is we have to choose the plant. So that's why we chose the plant depending upon the Ayurvedic knowledge, depending upon the medicinal properties, the phytochemistry of the plant. Now, what is this plant? This plant, Dioscorea bulbifera, it is a monocotyledonous dioecious plant. It can climb at a very faster rate. It can grow. It's a climber. It can grow at a very faster rate. That is 15 centimeter per day. You can imagine 15 centimeter per day. In one day, this plant can grow. In the month of June or July, there is bulbil or tuber formation at the base of the leaf. And then in the month of September, there are flower formation. So these flower formations, although there is flower, but these are sterile flower. They cannot give rise, they cannot pollinate, they cannot give rise to fruit. So these bulbils are the vegetatively reproductive organ. So the plant reproduces by these bulbils. Now in the month of November, there is a dieback mechanism. The plant dies. Even if you give water, you give fertilizer, you give all favorable condition, the plant will die in the month of November. So what remains on the ground is these Dioscorea bulbifera tuber. And this can remain viable for 1 year. They can remain dormant for 1 year. And after 1 year, in the month of March, when again they get some favorable condition, these Dioscorea bulbifera bulbs, they will sprout into new plants. So this is what is the life cycle of Dioscorea bulbifera. So what we choose is generally these tubers. These tubers have lot of medicinal properties. So we choose generally these tubers. Now why these tubers? Why the plants? Because we can use certain chemicals also for reduction and capping or stabilization. But these chemicals are very hazardous, they are toxic. So once this chemical attaches, suppose this toxic chemical attaches around the nanoparticle, then what happens? We cannot use the nanoparticle for nanomedicine purpose, then it will show toxicity. That's why we generally switch to some green method. Now, among different green methods, why Dioscorea bulbifera? Dioscorea bulbifera has a very rich phenolic and flavonoid content, like quercetin and others. At the same time, it has ascorbic acid and citric acid. So all these four components, phenolics, flavonoids, ascorbic acid, and citric acid, they act as reducing agent. They can reduce metal ions into metal nanoparticles. At the same time, the Dioscorea bulbifera has lot of starch and saponin. So during fasting, upvas, during fasting, the people of Johar, those in those places, the tribal belt, they eat this plant and they do not feel hungry for 2 days. You can imagine 2 days, like 48 hours, they do not feel hungry. Why? Because there is slow and sustained release of starch from these bulbs. So since these plants have a lot of starch and saponin, they can act as stabilizing agent. They can act as capping agent. Next, since we are not adding any other chemical, any other reducing agent or capping agent from outside, it's everything from the Dioscorea bulbifera, so it is more biocompatible. Only plant, green plant, it is edible. People are eating them. People are not getting diabetes. People are not getting cancer by eating this plant. So it is more biocompatible, more medicinal values are there. That's why we prefer this plant. Next, there is activity enhancement. Why? Because suppose Dioscorea bulbifera can prepare metal A nanoparticle. It can also prepare metal B nanoparticle. So the question is, can they form metal AB together, bimetallic nanoparticle, ABC trimetallic nanoparticle? So if they can produce these multimetallic nanoparticles, they can have much more effectivity, more synergistic activity. So what we are doing in our lab, we take different types of plants. So some of them I'll be discussing today. We take different parts of those plants, then we extract. After extracting, we synthesize nanoparticles. Next, we optimize different conditions, time, temperature, concentration, pH, and other things. Standardize and characterize these nanoparticles. Check their antimicrobial, antibiofilm, antiviral, and different types of biological applications. Next, we try to extract them, then isolate the important bioactive principles, characterize them, and then find out the mechanism of synthesis. So this is what we are doing. So that's why we can have some process patents and product patents also because of this whole process development. So what we have done over here, we have first tried to synthesize gold nanoparticle because Dioscorea bulbifera or Varahikand is known for synthesis of Swarna Bhasma during the earlier days in Ayurvedic medicine. So since it can be used for, even if you check the Dabur Sona Chandi Chawanprash, Dioscorea bulbifera is an integral part of that Chawanprash. Just see the label of that bottle and you will find Dioscorea bulbifera or Varahikand over there. Now, if you are looking at this plant, then we have taken these bulbs, cut it into pieces, then made powder and prepared extract. Before you synthesize a nanoparticle, before you start the synthesis, we need to know what are the chemicals that are present in the extract. So we found there is phenolic content, there is starch, there is reducing sugar, there is diosgenin, ascorbic acid, and citric acid. So you can imagine so many things are present in that single extract. So why can't we use that for synthesis of nanoparticles? That's why we started reacting with gold chloride solution, and it was one of the fastest method of synthesis. People take 5 hours, 6 hours, 1 day, but here the gold nanoparticle was synthesized within 90 minutes. So this is UV visible spectra. So the UV visible spectra showed a peak that was developed continuously from 5 minutes, 10 minutes, 20 minutes, till 90 minutes at 540 nanometer. And this 540 nanometer is the signature peak. It is the signature peak of gold nanoparticle. So this was a confirmation that gold nanoparticle might be synthesized within 90 minutes. Now we wanted to see what are the parameters that can impact or influence the synthesis. So here we varied the concentration of gold salt from 0.3 millimolar to 5 millimolar. And when we are varying the concentration, to our surprise, we saw the gold, same plant extract, remember, same gold salt, just changing the concentration, colors were different. At 0.3 millimolar, they were gray in color. Then it became red in color. Then it became ruby red in color. Then orange in color. And then finally yellow, like gold, golden color. So that means same gold salt, same plant extract, we change the concentration, we get different size of nanoparticles. As the size of nanoparticle changes, the color also changes. So we found that 1 millimolar was best in synthesizing gold nanoparticle. It was highest. Similarly, we varied the temperature 4 to 50 degrees Celsius. And we found 50 degrees Celsius was best. So 1 millimolar concentration of chloroauric acid and 50 degrees Celsius was most prominent, most effective in synthesizing these gold nanoparticles. Next, we wanted to see what is the shape, what is the size of these gold nanoparticles. So we found that these gold nanoparticles initially that were formed were very small spherical nanoparticles. They were coming to form smaller triangles. Now they were coming to form, aggregating to form bigger triangles. So there is a shape evolution. Very small to bigger and then much bigger. Similarly, there were smaller spherical nanoparticles, triangular nanoparticles, and hexagonal nanoparticles. So this is FESEM, field emission scanning electron microscopy. Next, we wanted to see whether it is really gold or not. So there is a method called EDS, energy dispersive spectra, that will give you which element is present. So we got the peaks of AU. You can see AU. So that means these particles were nothing but gold. So they were gold nanoparticles. What was the diameter of the nanoparticle? So there is a process called dynamic light scattering. Dynamic light scattering, DLS. And we found that there are very small nanoparticles around 11 to 30 nanometer over here. And there are some bigger nanoparticles, 50 to 300 nanometers over here. So you can see what we could see in the ACM, they were matching with the DLS, dynamic light scattering. Next, another method is there to see the purity and crystallinity of the gold nanoparticle, that is XRD. And this XRD spectra was also giving signature peaks of gold nanoparticle. We wanted to find out which functional groups are important in synthesizing the gold nanoparticles. And we found OH. This OH group was playing a very important role because OH is present, hydroxyl group is present in phenolics and flavonoids. So these OH groups were decreasing. Can you see over here? There is black. This black peak is more in the extract. Then when the synthesis was on, then it reduced to this red line, then it further reduced to this blue line. So there's a change. Slowly this OH peak is disappearing. And this carbonyl peak at 1718, it was being produced. If you can see, the carbonyl peak was coming over here. And this means that these groups are responsible for the synthesis. Next, we thought, if this Diascorea bulbifera can prepare the gold nanoparticle, can it prepare silver nanoparticle? In literature, in Ayurvedic literature, it is written that Diascorea bulbifera or Barahitan can prepare Rajat Bhasma also. So we thought, can it prepare silver nanoparticle? The Bhasmas are very, very small, biologically transformed form of metals, very small. So we thought, can it produce these silver nanoparticles? And within 5 hours, 300 minutes, within 5 hours, the silver nanoparticle was produced. You can see this is silver nitrate solution, and this is the silver nanoparticle that is brown in color. Here we confirmed, we tried to optimize the parameters, and we found that 0.7 millimolar was best. In gold, it was 1 millimolar, but for silver, 0.7 millimolar was best in synthesizing the silver nanoparticle. 50 degrees was best. It was showing highest nanoparticle formation. And we did HRTEM, high resolution transmission electron microscopy, as these nanoparticles are not bigger. These nanoparticles are not big like gold. So we found that there are very, very small spherical nanoparticles, very small, which are around 10 nanometers. And we confirmed with EDS that there was indeed silver. Hydrodynamic size also confirmed that 8 to 20 nanometer was majority over here, 8 to 20 nanometer silver nanoparticles, but they were coming together, as you can see over here. They were coming together to form bigger particles, which were very less, around 75 nanometer. We confirmed with the help of XRD its crystallinity and also FTI. Now we wanted to see what is its role in developing nanomedicine. So here we tried to check its activity against various bacteria, 14 different bacteria. So these are 14 different bacteria we checked silver nanoparticle. And we found at 9 microgram, you can imagine such less concentration, 9 microgram, it is able to inhibit E. coli, Escherichia coli. At 8 microgram, it was able to inhibit Klebsiella pneumoniae. At 10 microgram, it was able to inhibit Neisseria mucosal. So with almost at 20 microgram and 10 microgram. These are the zone diameter, 9 mm, 8 mm, and 10 mm zone diameter. So at 20 microgram or 10 microgram, it was able to inhibit. Such low concentration was so potent. Similarly, for Bacillus subtilis, you can see a zone diameter of 9 mm, you can see at 20 microgram over here. So we created one of the biggest library from this data, from this observation. We created one of the biggest library in 2012, and that's why this paper has got more than 600 citations in recent year. So this library had 23 different antibiotics. It is a combination 23 different antibiotics, 14 different bacteria, 14 different bacteria. And you can see streptomycin with silver nanoparticle, only streptomycin over here showing 7 mm zone diameter. A is only antibiotic. B, streptomycin plus silver nanoparticle 25 mm diameter. C, what is the fold increase? It is not times, fold increase. So 11.8 fold increase of this streptomycin. You add silver nanoparticle and streptomycin becomes 11.8 fold more powerful. You add silver nanoparticle, piperacillin becomes 3.6 fold powerful. Silver nanoparticle over here, it becomes 4.2 fold potent. It becomes 14.0 fold potent. So there is very high fold increase in the antibiotics when you supplement that, when you add silver into that. Similarly, like over here. So it was one of the very big library that we generated. And many people are referring this library. They are trying to develop different combinations of silver nanoparticle and antibiotic, and they are trying to develop nanomedicine from this library. Next, the question was, since it is able to synthesize silver, since it is able to synthesize gold, can we try something together? And when we added this, then it could show a peak that is blue, that is for gold. Then silver was showing this green peak. But when we added both, it was showing a different peak, very different peak, this red one. So this peak is not merging with silver, neither it is merging with silver, gold, nor it is merging with silver. So that means something different is formed. And 5 hours we synthesized, and at 5 hours the synthesis was complete. So at this point, we knew that when we combine silver and gold, it is not a mixture of only silver nanoparticle and only gold nanoparticle. It's not like that. Something different is getting formed. So what is this something? We did another microscopy, that is HRTM, high resolution transmission electron microscopy. And we found something was in the center and something was in the periphery, something was outside. But at this point, we did not know what was this something, what was inside, what was outside. So there is a process called elemental mapping. In elemental mapping, what we do, we give color code to different element. So here we have given green color to silver and we have given red color to gold. And we could see red was masked by green color. So that means silver was outside and gold was inside. So it is called gold core silver shell nanoparticle. And we confirmed that with the help of EDS, it was silver that was present as well as gold that was present in this. So both gold and silver were present in the nanoparticle. It is like our 10 rupees coin. Gold inside, silver outside, like our 10 rupees coin. Right? So over here you can see this gold core was 9 nanometer and silver shell was 15 nanometer. Very, very small nanoparticle. Now we thought bacteria cannot cause a disease alone. So if you take any disease like cystic fibrosis, periodontal disease, acne, any kind of disease, all diseases are caused mostly by the biofilms or multiple bacteria. When the bacteria come together, either they can be consortium or biofilm, So they form together, they form biofilm, and then they cause the disease. And these biofilms are very, very difficult to treat. So we wanted to see, does this particular nanoparticle have any anti-biofilm property? And we saw that when we were when we are treating the biofilm Acinetobacter baumannii, it could be inhibited up to 83%, more than 83% inhibition of Acinetobacter baumannii biofilm. They cause burn wound infection. Acinetobacter baumannii, it causes burn wound infection. If a person is uh, having like 90% burn or 80% burn, they do not have skin. And then Acinetobacter baumannii, Pseudomonas aeruginosa, they form biofilm. Then they form pus. And because of sepsis, these patients die. So this nanoparticle could inhibit Acinetobacter baumannii more than 83%. It could inhibit Pseudomonas aeruginosa more than 18%. It could inhibit E. coli up to 22% and S. aureus or Staphylococcus aureus 30%. So it was very important. Now we wanted to see what is happening inside the biofilm. So here you can see scanning electron microscopy of untreated biofilm of Acinetobacter baumannii. The Acinetobacter baumannii biofilm was completely intact. You can see the whole surface was covered with Acinetobacter baumannii cells, biofilms. As soon as we added our nanoparticle within 48 hours, how many cells were there? Only 6 cells were alive. Only 6 cells were there on the substrate. The entire biofilm was removed, eradicated. In case of Pseudomonas, you can see very intact Pseudomonas aeruginosa biofilm. When we treated with our nanoparticle, gold core silver shell nanoparticle, you can see only 2 cells were left. Pore formation was there, cell wall degradation, and the inner contents were coming out. So complete degradation of the Pseudomonas biofilm forming cells. E. coli, intact biofilm over here in C. Only one E. coli cell was left after treatment. You can imagine how potent, how important, how significant this nanoparticle is, bimetallic nanoparticle. This is Staphylococcus aureus, very intact Staphylococcus aureus biofilm. We treated with our nanoparticle. You can see the cell wall damage. It is coming out. The inner contents are all coming out over here. So there is complete damage. And you can see the exposure of the surface. The biofilm is eradicated. It is white. We wanted to confirm with the help of atomic force microscopy. And in case of atomic force microscopy also, we get 3-dimensional images. You can see Acinetobacter baumannii. After treatment, only 1 cell was left. Pseudomonas. This entire plate was covered with Pseudomonas biofilm. Only few cells are left. E. coli. Only 2 cells are left. You can see the nanoparticles, the crystals, nanocrystals on the substrate. Staphylococcus aureus. You can see over here in Staphylococcus aureus. Only patches of staphylococcal biofilms are left. You can see this substrate is exposed after treatment. Now we check another disease called Leishmaniasis or Kala Azar. Kala Azar. So Leishmania donovani is the causative agent. This is the Leishmania donovani cell. When we reacted with our nanoparticle, gold core silver shell nanoparticle, for 24 hours, there is complete breakdown of cytoskeleton. The entire cell was crippled. There was breakdown of cytoskeleton and it becomes spherical in shape. You see, this is a spindle shape and this is spherical shape. Here, we did AFM and you can see the spindle shape, very intact Leishmania donovani cell. After adding the nanoparticle, you can see the cytoskeleton has collapsed. This is the DNA, intact DNA of Leishmania donovani. After reacting or treating with the nanoparticle, there is smearing of the DNA. So this gave us very significant result that it can be anti-infectious. So we can develop some anti-infectious agent from these nanoparticles. Now coming to cancer, cancer has become such a difficult disease to maintain. Means there is no answer to cancer till date. We are developing so many medicines, but you can see so many different types of cancers are there. The medicines that are used nowadays are cisplatin, carboplatin, oxaliplatin, pyriplatin. But the problem is these drugs which are used in chemotherapy, they do not know which one is healthy cell and which one is cancerous cell. So they go and they can kill the healthy cells also. So there is lot of side effects, adverse effects because of cancer chemotherapy. That's why we thought, can we develop these platinum based? You can see these drugs are platinum based. Cisplatin, carboplatin, oxaliplatin, platinum based. So we thought, can we develop platinum nanoparticles, platinum based drugs from this plant? So we used the same Diascorea plant and we took the extract. And after using this extract, we saw that it could produce platinum nanoparticles. What is the size? Just 2 to 5 nanometer, very, very, very small nanoparticle. The smaller the nanoparticle, better the activity. It could produce palladium nanoparticles, which was 10 nanometer. It could produce platinum palladium bimetallic nanoparticle, which was 25 nanometers. So these are very, very small nanoparticles. You can see the color change when the platinum nanoparticles were formed. This is platinum chloride. And after platinum nanoparticles are formed, this is the color. This is palladium nanoparticle, black in color. Platinum palladium, intense black. So we checked EDS and then did the anti-cancer activity, tried to prove it, and then we did antioxidant activity, check the antioxidant activity. So we found those nanoparticles were indeed made up of platinum. See, this is the platinum nanoparticle, and you can see the PT peaks, platinum peaks. This is the palladium nanoparticle, you can see the PD peaks. And in this, you can see both PT and PD. PT and PD. So that means these nanoparticles are having both the elements. Again, FTIR, we tried to find out which groups are playing role in synthesis and stabilization. So CH, NH, NO. So these groups were playing important role. Now, in case of anti-cancer, we used HeLa cell line, cervical cancer cell line. When we used platinum nanoparticle, there was around 15% cell death, HeLa cell death, cancer cell death. When we use palladium nanoparticle, here we could see around 32% cell death. When platinum palladium together, it is around 75%. You can see it increased. Platinum less, palladium less, but together it was very high. And we used a control standard drug cisplatin, it was also similar. So our platinum palladium bimetallic nanoparticle was able to show similar activity as the standard drug. We checked the flow cytometric analysis, whether it is inducing apoptosis or not. So it is by dual stain, annexin 5 and PI, propydium iodide. So if annexin 5 and propydium iodide are present together, that is called apoptosis. So we found with platinum, 15% cells were undergoing apoptosis. With palladium, 10.51% cells were undergoing apoptosis. But when we are combining together, then you can see in this quadrant, 20.6% cells were undergoing apoptosis, program cell death. So what is happening? How do we use these two types of stains to see the apoptosis? Here you can see the HeLa cells. Now, if there is damage, if the cells are dying, cells can die by two methods. Either it can die by apoptosis, programmed cell death, or the cells can die by necrosis. So, when there is damage due to either apoptosis or necrosis in the cell membrane, then PI can go inside. So, PI tells there is damage in the cell membrane, either necrosis or apoptosis. Here it is healthy cell. So you can see there are no PI almost. Some cells which are dying because of normal. Every cell has a lifespan. So they will die because of their normal death. But here very less. Here you can see PI is more. That means cells are dying. Now, annexin 5 FITC. FITC is a fluorescent dye which is bound to annexin 5. Annexin 5 can bind to phosphatidylserine. Phosphatidylserine comes out in the outer membrane only during apoptosis. So, if there is apoptosis, phosphatidylserine will come out. If there is phosphatidylserine in the outer part, there will be binding of annexin 5 over there. If there is binding of annexin 5, the FITC will show more green color. So more the green color, more the apoptosis. So here you can see the dual staining apoptosis over here. Here also you can see apoptosis when we treat with palladium. And when we treat with platinum palladium together, you have more very high amount of apoptosis over here. Now we saw the antioxidant activity also, because generally the cancer is caused by free radicals, ROS, reactive oxygen species, oxidative stress. So we wanted to see what is its effect when we are adding these nanoparticles. So when we are adding platinum, palladium and together, we could see bimetallic nanoparticle were scavenging this DPPH radical more. Bimetallic nanoparticle was showing highest superoxide radical scavenging activity, highest nitric oxide radical scavenging activity, and very high hydroxyl radical scavenging activity. So this meant this bimetallic nanoparticle is very good free radical scavenger and can protect from oxidative stress. Now the question is, it is showing anti-cancer effect, no problem. Now how can we target? Can we develop some nanomedicine for targeted delivery? So first we saw against infection, next we saw against cancer. Now we are going for targeted delivery. So for targeting, we used iron oxide core nanoparticle. So this iron oxide nanoparticle functioned as the core. Then we wanted to add a bioactive principle called diosgenin. This diosgenin is an anti-cancer bioactive principle of Dioscorea bulbifera. It is a saponin. Now, when we wanted to add this diosgenin on this core, we needed some linker, some bridge. So citric acid was working as the bridge. Now, why we selected this? Iron oxide has iron, even our hemoglobin has iron, so it is non-toxic. Citric acid, we find in citric acid cycle, TCA cycle in our respiration, in all cells. So citric acid is already a metabolite of our body. And diosgenin, we get in methi, fenugreek. Diosgenin, we get in Dioscorea. Diosgenin is an edible saponin. So all these components are biocompatible. That's why we thought of using this. Now, this diosgenin is such an important component. It knows it can go and kill only the cancer cell and it will not kill the normal cell. The size of these nanoparticles range from 12 to 21 nanometer, around 12 to 21 nanometer. So you can see this is bare iron oxide nanoparticle. This is citric acid coated iron oxide nanoparticle. This is diosgenin functionalized citric acid coated iron oxide nanoparticle. We did the HRTM analysis similarly and we found these structures. Now, why they are together, why they are not free? Because they are magnetic. They attract each other. That's why they are always in clusters. So we saw the hydrodynamic size of the clusters, and only iron oxide nanoparticle were around 65 nanometer, the clusters. Iron oxide nanoparticle with citric acid were around 119 nanometer, the cluster size. Iron oxide nanoparticles with citric acid and diosgenin, they are around 145 nanometer. We confirmed that whether diosgenin was present on that iron oxide or not by FTIR. This red color is the pure diosgenin, and this blue color is iron oxide nanoparticle with diosgenin. So we can see the same peaks of diosgenin over here. This confirms that diosgenin is present in the nanoparticle. We saw the anti-cancer effect against MCF7. This is breast cancer cell line. And we found that iron oxide, diosgenin functionalized iron oxide was giving highest activity, around 50% as compared to others. Now, there is a very significant study that we conducted. Initially, cancer, before forming a cancer, there is a benign tumor. In one particular site, there are the tumor cells. So that is called benign tumor. It does not, the cells, they do not dislodge, they cannot move to other parts. But slowly, these cells will become malignant. They will start coming out from the tumor site and they will move at different parts. So it is called metastasis. When it starts metastasis, when there is migration of these cells, then the cancer spreads in the whole body, in different organs. So our aim was not only to inhibit cancer, but also to inhibit the migration of the cancer cell. So what we did over here, it is called wound migration assay. Here you can see a control. What we are doing in control? We are having a lawn of MCF7 breast cancer cell. And on this lawn of MCF7 breast cancer cell, we created a wound, a gap on all cells, we created a gap. So in the control, which was untreated, after 24 hours, the gap was almost sealed. Why the gap was sealed? Because the cells from different parts, from both the sides, they were coming together, sealing the gap. Here with iron oxide nanoparticle, the gap was almost sealed. With iron oxide citric acid, the gap was less sealed as compared to this. But with iron oxide diosgenin, the gap was not sealed. That means the migration of the cells was inhibited by this. Similarly, with only citric acid, there was less migration. With diosgenin also, there was less migration. So iron oxide, which was functionalized with diosgenin, it was showing more inhibition of wound migration. You can see diosgenin functionalized iron oxide nanoparticle only showed 40% migration. Iron oxide nanoparticles showed around 89% migration. And iron oxide nanoparticle citric acid showed 50% migration. So the lowest migration was in diosgenin functionalized iron oxide nanoparticle. You can see over here. Lowest migration. So it was very efficient. Now we tried to see whether it is apoptosis or necrosis, and you can see in iron oxide diosgenin, same. We can see maximum, both the green also and red also. The propydium iodide staining was also here, and Alexaflor 488. It is similar to the previous dye, like Annexin 5, Alexaflor or FITC. It is similar. Alexaflor is similar to Annexin 5 FITC. So it also stains the phosphatidylserine. So you can see dual staining positive over here. So this confirmed that it was undergoing apoptosis. Similarly, so we saw infectious disease, then diabetes, then cancer, and now we are coming to diabetes. In diabetes, we generated copper nanoparticle. In our earlier days, people used to keep water in copper vessels. And when they used to drink that water next day morning, they were not getting diabetes because very, very small copper particles used to leach out in the water. And drinking that copper leached water helped to cure diabetes or prevent diabetes, protect from diabetes. So what we did, we tried to see whether this plant can synthesize copper nanoparticles or not. Within 5 hours, copper nanoparticles were synthesized. You can see very nicely spherical copper nanoparticles around 12 to 16 nanometer. We confirmed with the help of EDS and we checked the enzyme inhibition. This is the amylase. Now, why we are getting blood sugar, more blood glucose? Because when we are eating some starch containing food, the starch is initially degraded to maltose, isomaltose, or maltodextrin, and then after degradation, so this is done by amylase. After degradation of this, then further smaller sugars are degraded by glucosidase enzyme to glucose, and that glucose comes in blood. It increases the blood glucose. So the question is, can we inhibit this enzyme? If we inhibit this enzyme, alpha amylase, alpha glucosidase, then probably it can stop the metabolism of the sugar, and it will stop the increase of blood sugar in diabetic patients. So we found that this copper nanoparticle was showing inhibition of porcine pancreatic alpha amylase, murine pancreatic alpha amylase, and murine intestinal alpha amylase. And then we found out the mode of inhibition. This is the fluorometric analysis. So the tryptophan, tyrosine, the amino acids of the enzyme gives this fluorescence. When the nanoparticles were added, the fluorescence slowly decreased. That means the nanoparticle was going and changing or altering those amino acids. We checked with the help of circular dichroism, CD spectra. CD spectra gives you the structure, conformation. So there was a conformational change. You can see the black one was untreated enzyme. When the nanoparticle was added, the spectra changed to the red one. And we confirmed with the help of docking study that it is binding to tyrosine, tryptophan, and phenylalanine, the main aromatic amino acid. Similarly, we found the alpha glucosidase around 99% alpha glucosidase inhibition. It was similar to acarbose drug, standard drug, 99%. Urine intestinal glucosidase around 90%, more than the standard drug. It could also scavenge different free radicals like DPPH, nitric oxide, and superoxide, and we confirmed with the help of ESR spectrum. So this was about the anti-diabetic study. Now, a very important, we are coming to the end of our talk. Now, a very important observation. Now, since you are, most of the people are from audience, are from means Maharashtra and different places, people from Maharashtra can identify this. So this is the Western Ghat of Maharashtra. Now, in Western Ghat of Maharashtra, as we know, in India, there are 12 Jyotirling. There is one Jyotirling which is called Bhima Shankar. So during my weekends, I used to go and visit these tribal belts, tribal areas at different places of Maharashtra. So when I visited Bhima Shankar, I found this plant, this tree. This is a shrub. And this tree, this particular plant grows at a height of 3,394 feet. You cannot grow this at a lower level. Only at high altitude, this plant grows. And the cattle, the cows, the buffaloes, they were maintaining 20 feet distance from this plant. So I was very surprised when I observed. I found the cows, buffaloes, they were not going near this plant and they were maintaining 20 feet distance. So I asked the Vaidus, the village doctors are called Vaidus. So I asked the Vaidus, why they are not going to this plant? They told, sir, if you smell this plant, all of your teeth will fall down. If you smell this flower of the plant, all of your teeth will fall down. So we call it Datpadi. It's called Datpadi plant. Also, it is called Rameta. So I asked, then is there any other beneficial effect? So what they do, every Monday, they have a village market. They take very thin and lean cows which are going to die. And then they make a paste of Datpadi leaf and put it on those thin cows. Cows, buffaloes, very thin cows, they put a paste, Datpadi paste, leaf paste. And because of the allergic reaction, there is swelling. And because of the swelling, the cows, the buffaloes, they swell up and they look very healthy. So they go to the market, they take them to the market and sell them at 80,000, 85,000. The person who is buying these cows and buffaloes, after 2 to 3 days, when the swelling subsides, they see the cow is so thin, it is going to die after some days. And they are cheated. The cow does not worth 8,000 also. And they brought that cow home at 80,000. So people are using the immunology, hypersensitive reaction to make money. We are learning immunology in our library, in our textbook, QB, Royd, different types of immunological reactions. But see, people at Bhimashankar, they are making money from immunology. They are cheating by using hypersensitive reactions, allergic reactions. So it was very interesting. And this plant, you can see, this Natpadi flower, it has so many different types of medicinal properties. It is anti-rabies also. Against dog bite, this Natpadi flower can be used. So we synthesize the gold nanoparticles, and you can see intact triangular and hexagonal gold nanoparticles were seen. This is elemental mapping. So here, very interestingly, an important observation we found that initially very small particles were found. They were coming together to form bigger particles, and they were coming together to form triangles. This is called Oswald ripening process. It is theoretically explained, but it is very rare to see in practical application. But Dathpadi leaf, Nidia glauca leaf actually showed this to us. So initially very small, 12.3 nanometer, very small nanoparticles were formed. They were coming to form bigger nanoparticles. They were 59.46 nanometer. They are coming to form much bigger triangles around 286 nanometer. Oswald ripening process. So we thought, can we modify the process over here? Can we control the shape? So as soon as we added Nagpari leaf, that is the Nidia glauca leaf extract to gold chloride, there's nucleation. Very small particles are formed. Now these particles come together to form different size and shape. At this point, we added chloride ion. When we added chloride ion, we got nano triangles. When we added bromide ions, we got nanopentagons. When we added iodide ions, we got nanowires. When we added fluoride ions, we got nanospheres. So by changing the halide ions, we could change the shape, we could change the morphology of the nanoparticles. So likewise, we have worked on different other components like curcumin, haldi, turmeric. Turmeric has curcumin. Curcumin can cure so many different types of cancer. A single molecule of curcumin, you can imagine, just curcumin molecule can cure so many different types of cancer, can inhibit so many different types of cancer. How? One molecule of curcumin, just curcumin molecule, can control so many gene expression. So these brown are cancer causing genes. So they are going to downregulate so many cancer causing genes. They will downregulate. They will stop the expression. Curcumin can upregulate the anti-cancer gene, the blue ones, which stops cancer. These type of genes, curcumin upregulate the expression. So you can imagine how important haldi is. And US USA now prepares this haldi capsule because their pizza, their burger, the Western food, they do not have turmeric, haldi. We have turmeric in everything, right? From morning poha till varan, in any kind of Indian food, we have turmeric. But in US, they are now using these haldi capsules. So you can see, these are the haldi capsules. These are the small haldi capsules they are using in order to prevent cancer, diabetes, and different oxidative stress related disease. And we are shifting to Western diets. We like more pizza, we like more burger, which are without any kind of curcumin. But Indian food is so important medicinally. So we conjugated this curcumin onto iron oxide nanoparticles with citric acid bridge, and the size was 15 to 20 nanometer. It could inhibit the tumor up to 90%. 90% tumor inhibition, curcumin, when it was conjugated to iron oxide. And it could inhibit or it could scavenge the free radicals also. You can see superoxide, 100% free radical scavenging over here. 100%. So potent this compound was. So in conclusion, these nanoparticles, these biogenic nanoparticles are very efficient and they can be produced environmentally by environmentally benign route. These nanoparticles can inhibit biofilm, they can inhibit cancer, diabetes, and lot more different diseases can be targeted. And if you're looking at this nanotechnology, very recently we come over the COVID-19. Now see, when SARS-CoV came in 2002, 2003, 8,000 people were infected. Among them, 800 deaths were there. When MERS-CoV came, this coronavirus in 2012, infected people were less, but deaths were 858. So the virulence was more, you can see, 8,800. And here, 2,494 and 858. So MERS-CoV was more potent, more dangerous. And in late 2019, when COVID or the SARS-CoV-2 came, you can imagine, you can see how many deaths. 70 lakh, 10,681 deaths till January. Now again and again, there are new cases. So you can see so many deaths and so many infected. So at that time, people started thinking what we can do, because the virus is at 120 nanometer size range. If we want to develop some nanomedicine, if we want to develop some nanoparticle, we have to cut down to that range of the virus. The medicine should be at that range. And that's why people started producing different types of nanoparticles which were supposed to bind to this spike glycoprotein and this envelope proteins over here. So Israel Sonovia, they created zinc oxide nanoparticles and put them in fabrics, in textile. So whenever the virus was trying to attack, it could bind to this zinc oxide nanoparticles and it was denatured. So there are different nanoparticles are there. So these are some of the books that we have published. We are still publishing different books on nanobiotechnology. See, Diascorea bulbifera is one plant. It is a model system of nanobiotechnology. One single plant, Diascorea bulbifera. It can synthesize silver, gold, copper, platinum, palladium, so many different types of nanoparticles. So these are different. So nanobiotechnology, agriculture, in medicine, biofilms, and different types of other nanoparticles also. So these are the books that we have published. So I am very thankful to Dr. Amrita, principal and convener of this conference, to Dr. Pingle, the co-convener, for inviting me, all organizers for organizing such a wonderful e-FDP and e-STP that will benefit all, and all participants for listening. All our researches are funded by different agencies like Northeastern University, USA, Kasetsart University, Stanford University, then DBT and DST, CSIR. And these are the students who have worked on these particular areas which I just explained. Thank you so much. I'll be happy to take any questions if there are.
Thank you, sir. Thank you very much. Sir, some questions from audience.
Yes.
That first question is, why the size and shape of nanoparticle changes uh, when added the different halides?
Yes. The main thing is when we are adding different halides, the size of the halide atoms, they are different. If the size of the halide atoms are different, they act as template. So they help in aggregation of these nuclei particles on their surface, and they actually help as template in changing the size and shape of the nanoparticles. There are very important papers on in Langmuir that was published in 2012. So at different time points, the people are trying to develop, but we found that if people have reported chloride ions for triangles and some, but then we thought, can we vary all halide ions? Can you take all halide ions? And we found that it depends upon the shape, the size of the halide atoms that act as a template in changing the size of the nanoparticle and shape of the nanoparticle.
Okay, thank you. One more question, sir. What are the challenges and limitation of using nanoparticles as a green approach, and how can they address?
Pardon, which approach?
What are the challenges and limitations of using the nanoparticles as a green approach?
Okay. Yes. So the main challenge is you have to note down the size. The nanoparticles which are below 80 nanometers, 80 nanometer, below 80 nanometer, they can be easily cleared out through our excretory system, through urine. If the nanoparticle is bigger, then probably there will be more problem of bioaccumulation. It can be accumulated in different organs and there will be toxicity. So the smaller the size, better will be its application.
Okay, thank you. One more question, sir. What are the potential economic and social benefits of using the nanoparticle as a green approach?
Yes, so now you can see the COVID vaccine. They were liposomes, nanoliposomes, the Moderna vaccine. So now people are coming down to the nano form. So there is a lot of economic importance. Even if you go to the chemist shop, there is silver sulfur diazine cream. You go to any chemist shop and you ask that I have got a burn infection. Previously we had burn on, but now we have silver sulfur diazine cream. So very, very small nano means silver is added, mostly like silver nitrate people are adding, but the silver nitrate is sometimes toxic. So you can develop such kind of application. Being pharmacists, being the students of pharmacy or the researchers from pharmacy background, there is a lot of scope that you try to develop formulations. When these formulations are developed, they can be very, very useful. Now people are not changing drug like amoxicillin. Bacteria is getting resistant against amoxicillin. So people are what they are doing, they are generating nano-amoxicillin, nano-antibiotics. So it is called nanobiotics. No more antibiotics. Now people are shifting to nanobiotics. So if those antibiotics are very, 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 very small form, then they can penetrate within the cell. They can have more therapeutic potential. And it is the same antibiotic. So you do not have to take again FDA approval. You are just changing its form from the bulk microform to nanoform. You are not changing the drug. You are just changing its form. So there is a lot of economical benefit if you can convert the pre-existing drug into nanoforms.
Thank you. Thank you, sir. Ladies and gentlemen, esteemed speakers, participant, I am honored to conclude the key takes away from our session on nanoparticles and integrated interdisciplinary green approach. Today's discussion highlighted the vast potential of nanoparticles in addressing various environmental and industrial challenges. Sir emphasized the importance of integrated and interdisciplinary approach combining the concepts from various topics. Thank you, sir, uh, for his valuable talk on nanoparticles and integrated and interdisciplinary green approach. I am also thanks to all the participants and organizers for making this session to a success. On behalf of Gokhale Education Society, Sir Dr. M.S. Gosavi, College of Pharmaceutical Education and Research, Nasik, principal and staff, so please kindly accept the certificate of appreciation. Thank
Thank
you.
you so much.
Thank you very much. Thank you.